Okay, so now we're going to talk about genetic diversity. This is the sorting and recombination of genes in sexual reproduction that results in a great variety of gene combinations in the offspring of two parents. Do you um, exactly like your do you look exactly like your brothers and sisters? So because of the recombination of genes that occurs during meiosis, we have genetic diversity. Genetically diverse populations are more likely to survive changing environments. Greater variation within the species makes a population better suited to adapt to changes in the environment. And all that means is we have more diversity, um, such as uh, birds with different beak sizes that would have more that would lead to more genetic diversity. We'll say, this population of birds that have all these different beak sizes can only eat grass seeds at one point. Well, if a drought comes along and it kills the grass and there are no more grass seeds to eat, they could likely move on to another food source. The ones that have bigger beaks could. Whereas the ones that do not would likely die. Genetic diversity. So, leopard populations around the world are in danger because of inbreeding, because there's not a lot of genetic diversity that leads to adaptation. There is very little genetic variation between any two individuals. This makes them very susceptible to disease and will likely lead to their extinction. Recombination and mutation provide for genetic diversity. So the inserting, deleting, or substituting of DNA bases can alter genes. An altered gene in a sex cell may be passed on to every cell that develops from it, causing an altered phenotype. Um, so pretty much what this means is we get genetic diversity through um, a couple of different ways. We get it through recombination. We get it through just and recombination is just what we were talking about with crossing over. We swap genes on different chromosomes. We also get it from joining egg and sperm. So we get, half our DNA from our mom, half our DNA from our dad. That gives us a little genetic diversity there. And then sometimes random mutations occur in the population, and that provides for genetic diversity. Not all mutations lead to um, uh, cancer or disease. Sometimes it leads to a new phenotype in the population. Whereas a good example of this is one time I was hiking in the woods, and I looked over and I saw a blue dog-day cicada. And I don't know if you've seen those before, but they're normally green. Now, if so, what had happened was more than likely a mutation had occurred in that individual uh, that caused it to produce a blue color instead of a green color like it normally is. And if that's an advantageous trait, then that will be passed on to other offspring. So crossing over, you should have learned about this last week. This is the physical exchange of chromosomal materials between chromatids of homologous chromosomes. The result is generation of new recombinations of genes. So it occurs in prophase and meiosis 1, and it generates diversity. So I'm just going to show you. That's what happens. It creates chromosomes with new combinations of alleles for genes A to F. So here we have an individual right now. If we took your chromosomes, we would have one of each chromosome. One would be from mom and one would be from dad. Okay, so let's say the yellow is from mom and the green is from dad. Well, during S phase, we know that this duplicates, so we get two exact copies, all right? So what happens in prophase 1 of meiosis when we're creating sex cells is letters, and this just says letters denote genes, the case denotes allele, and alleles have crossed over to produce new gene combinations. Okay, so alleles have crossed over to produce new gene combinations, okay? So what this showed us was these genes right here were originally on this sister chromatid, okay? And this whole thing right here is called a homologous or homolog um, pair of chromosomes. Well, what's going to happen here is during it goes through two divisions, and what's going to happen is you're going to end up with, let's say that these were chromosomes in a female. She's going to have one of these chromosomes sitting in 4 different egg cells. So here we have a duplicated maternal chromosome. Here we have a duplicated paternal chromosome. We have a sister chromatid. And then what it does is it forms, when we have the two like this, it forms a tetrad. And they're just kind of going to cross over each other and swap genes. Genetic diversity, sometimes entire chromosomes can be added or deleted, resulting in a genetic disorder such as trisomy 21 or Down syndrome. And you're going to learn about, this week, you're going to learn about karyotypes. And that's kind of a cool thing to learn about. Genetic diversity. So chromosomal errors such as non-disjunction, the failure of chromosomes to separate properly in meiosis. Gametes with extra or too few chromosomes result and can cause disease such as Down syndrome. So during what happens is during meiosis, instead of getting let's say, I'll go over here, instead of getting, we have 23 pairs of chromosomes here, something might happen and we get an extra chromosome here that should have been in another cell, and that other cell didn't get a chromosome, so they failed to separate like they should have. And that can cause Down syndrome and anything where you would have an extra deleted chromosome. We also have polyploidy, which organisms with entire extra sets of chromosomes results in the death of a fetus in animals, often occurs in plants and causes the fruits and flowers to be larger. So here we have what we call a karyotype. And this is just a picture of your chromosomes. It's really neat. We can actually freeze your cells in metaphase, and we can map, we can make a little map of them, judging by how long they are. We actually look at the banding of the banding patterns that occur, and you'll learn about this. There's a dye that can stain the chromosomes to where they create this banding pattern. And someone will actually sit at a computer and line these up so that we have a karyotype. And sometimes this is used to diagnose genetic diseases. Sometimes this is used to diagnose lymphoma, leukemia. They take a blood cell and they freeze it in metaphase, and they take the chromosomes and they line them up, and a lot of times you'll have tons and tons of extra chromosomes when something like that happens. If you look at this, you're going to notice that we do have 23 chromosomes. We have 1 through 22. These are our autosomal chromosomes. And then we have one set of sex chromosomes. And it's showing us here we have XY, which means we have a boy. Here we have a karyotype. And you can notice here we have 1 through 22 here. But we have an extra set right here, 1, 2, 3, which is chromosome 21. And we have an extra chromosome 21 because we're only supposed to have 2 here. And what happens here is this would be someone with Down syndrome. They have an extra chromosome 21. And if you look over to your right, you'll notice that it's XY, which tells us again that it's a boy. We also have Turner syndrome, and you'll learn about this too. Turner syndrome is a genetic disorder affecting only females in which the patient has one X chromosome in some or all cells, or has 2 X chromosomes but one is damaged. So it either has one X chromosome the patient has either one X chromosome or it has one that's damaged. Signs of Turner syndrome include short stature, delayed growth of the skeleton, shortened 4th and 5th fingers, broad chest, and sometimes heart abnormalities. Turner syndrome, women with Turner syndrome are usually infertile due to ovarian failure, and diagnosis is by a blood test or a karyotype. And here we have a karyotype showing us Turner syndrome. We have that missing X chromosome, which should be right here. It should be one that looks exactly like that. Huntington's disease. Huntington's disease is an inherited disorder caused by the degeneration of certain nerve cells in the brain. The gene for Huntington's disease is codominant. So HD causes bizarre and involuntary movements and loss of intellectual abilities. The condition begins most often in mid-adulthood and progresses slowly to death. And what happens is the brain just actually degenerates. The identification of codominant gene for HD now makes it possible to determine who will develop this disease by examining their DNA from a blood sample in the laboratory. Here we have Huntington's disease, and it's just showing you what we have here. We have a parent that has Huntington's disease that's passing on Huntington's disease, and you have to have the 2 alleles to actually have Huntington's disease. So that leaves you at a 50% risk to actually obtain Huntington's disease if your parents have it. Fragile X syndrome, inherited disorder caused by a defective gene on the X chromosome. Symptoms of Fragile X syndrome are mental retardation, enlarged testes, and facial abnormalities in males and mild or no effects in females. It is most common inherited cause of mental retardation. And here we have Fragile X syndrome, and it's just showing you that the gene that's located, the defective gene on the X chromosome. Okay, so Crudicat syndrome. This is, my students love to talk about this one. Crudicat syndrome is a rare genetic disorder due to a missing portion of the chromosome 5. Its name, meaning cat cry in French, is from the distinctive meowing sound made by infants with the disorder. The disorder is characterized by distinctive facial features, small head size, low birth weight, weak muscle tone, round face, epicanthal folds, low set ears, facial asymmetry. Severe mental retardation is typical. And here we have